All right, let's begin. Um, it's my pleasure to introduce Dennis Dean, who is going to give the next talk on nitrogenase. Dennis has worked on nitrogenase for many, many years. Dennis is a professor of biochemistry in the College of Agriculture and Life Sciences. And he has been a member, as far as I can tell, of Virginia Tech for at least 36 years. He went to Wabash College and Purdue University. He was an NIH postdoc at the University of Wisconsin and a staff scientist at a not really well-known group, which was very famous a long time ago, the Kettering Research Lab. I don't know if it still exists, does it?
No, no.
The Kettering Research Lab was a very famous lab for basic research in plant biology and photosynthesis and many things before he joined Virginia Tech in 1985. He is the founding director of the Virginia Tech Fralin Lab Life Sciences Institute, and he's a distinguished professor and the Strutzberg Professor of Biotechnology. So take it away.
Okay, thanks, Paul. I want to start by thanking Paul, and I also want to especially thank Beatrice for her help. I was just commenting to her that I'm a very nervous kind of person, and I'm very needy, and she's been very polite and very responsive, and I really appreciate it. It makes me really feel good. And if I was still an administrator, which I'm not, I would try to steal her to come work here. So what I'm going to talk about today is nitrogenase. And as you can see, there's a whole cast of people that we work with and interact on a very regular basis. One of those is Betul, who spoke yesterday and gave a fascinating talk. As you can see, though, I put down the expertise of some of these people. And so we have a real breadth of expertise and knowledge. And it's really delightful to have these individuals to work with. And I'd like to say we have a very lively interaction. And by lively, I mean we don't always agree on everything. And the conversations can get more than lively. But we're all very good friends and we're working together on what I think is one of the most important challenges facing us today, and that is the mechanism of nitrogen fixation and how we can use it for agronomic benefit. I also want to point out that I have 4 current colleagues in my group. When I graduated with my PhD, my advisor told me, Dennis, if you want to be successful, you have to be able to get people to work in your lab that are smarter than you. And I've been very successful in over my career, and this is a particularly talented group. I also want to mention the funding that's for our lab. It comes from the Department of Energy, the Basic Energy Sciences, and also the Bill and Melinda Gates Foundation. So what I'm going to do today is first talk a bit about the organism that we use as a model organism, and it's for nitrogen fixation. And it's one that many, many people now use. It's Azotobacter finlandii. Azotobacter meaning nitrogen-eating bacteria. And Paul probably knows that it was isolated in his backyard in New Jersey. The things about it that are nice is it's easy to grow. It's very genetically tractable. The genome is sequenced. It produces high levels of nitrogenase. It produces 3 kinds of nitrogenases, which I'll talk about. And mostly from the Reese laboratory, there's very, very good high resolution structures that are available that are very informative. So this is the Azotobacter here, a micrograph. You can think of it as Pseudomonas with nitrogen fixing genes. Here in the bottom left, you can see that nitrogenase is when the organism is fixing nitrogenase, it makes the catalytic components, NIF-D, NIF-K, and NIF-H, which I'll mention a bit in a few minutes, at just very, very high levels. It's up to about 30% of the total cellular protein. And the reason for this is that the enzyme is very slow. Another good aspect is because the system is genetically tractable, it's easy to do things like put affinity tags on the protein of choice, and it can be easily isolated by affinity chromatography methods. Here's a case of a crude extract. You can see These are the subunits of the catalytic component. This is the purification can take just a couple hours. In some cases, even less than that, to get very high yields. Also, referring to Batul's talk yesterday, the other really nice thing about this is this organism, it has an extremely efficient transformation system. Just whisper DNA and it takes it up and will incorporate it into its chromosome and an exchange, an allelic exchange. And that's very important because there's some, and as I'll discuss, there's so many components involved. It's not something you can just put on a plasmid and shove it in E. coli. There's a lot of different genes that have to work together in a synchronous and coordinated way to make the system. So you've got to do your genetics on the chromosome in the native host. And this just shows a very old gel from my colleague Barbara Burgess and I from a paper we published about 35 years ago. And this just shows these are the catalytic components here, all the other cellular proteins. And this is one where we've engineered taking those genes out. And if you can precisely take the genes out, you can play around with them in E. coli, and then you can put them back in, which is important for doing site-directed immunogenesis and also doing biochemical genetic studies to try to decipher who's doing what with whom and so on. The other thing that's nice about working on nitrogen fixation is so many of the proteins involved have a color to them. And that's really nice when you're trying to train undergraduates in the lab. They really get turned on by seeing the colorful proteins on the columns. It's an easy way to teach them about how to do column chromatography and protein purification and so forth. And so these are just a variety of the proteins that are involved, and I'll be mentioning a few of these. So here's the enzyme, and this shows a half of it. So actually the molybdenum iron protein is a tetramer, so you could see on this other side, if you're looking at the whole system, you would see this mirror image with this. And the iron protein is called an iron protein because it has a 4-iron 4-sulfur cluster. And it is responsible for the delivery of electrons to another cofactor mediated by another iron sulfur cluster. But the action really occurs on what's here is called M cluster or iron molybdenum cofactor. Now, the trick of all enzymes, of course, is to lower the activation energy. And you can see in this diagram here with reaction coordinates that overall, fixing nitrogen is a downhill process. But the problem, the kinetic problem, is getting it to a state where it's activated, which is a very unfavorable process. So once we can form the diazine intermediate, the reaction is very favorable. And so what I want to do now is go through some of the steps involved in electron transfer and substrate activation and so forth. And to do that, we need to consider, well, I'm going to back up here. So during the process of nitrogen fixation, these two proteins, the iron protein and molybdenum iron protein, associate and dissociate. And for each association and dissociation, there is a single electron transfer. And so the key to catalysis is accumulating enough electrons to get this activation. And the association and dissociation of these two catalytic partners involves the hydrolysis of two magnesium ATPs, which, although not shown here, are attached to the iron protein. Many people intuitively believe that it's ATP hydrolysis that drives electron transfer. We've recently shown that's probably not the case, but rather it's the energetics of the association of the two proteins and conformational charge changes, which leads to electron transfer. And then it's the dissociation of the two component proteins, which involves magnesium ATP hydrolysis and phosphate release. But let me now go through some of the key observational, experimental observations. And this is not from my lab, most of them, some are, but most of them are not. And these are the things that need to be satisfied when we start thinking about mechanism. And as Paul mentioned, this is an incredibly devious enzyme because it is so complicated. So let me go through these very quickly, and I'll try to explain them in the next slide. So electrons are delivered from the iron protein to the molybdenum iron protein one at a time. Acetylene, which is often used as a proxy for nitrogen reduction, can also be reduced by the enzyme by only 2 electrons to yield ethylene. Acetylene is a non-competitive inhibitor of N2 reduction, but N2 reduction is a competitive inhibitor of acetylene reduction. In the absence of all substrates, any substrate, all electrons are diverted to proton reduction. And this is an important point from the perspective of experimental analysis, is because it means the enzyme never sits still. If it accumulates electrons, and it's always, if it's not reducing substrate, it's always going backwards. And so it's very difficult to trap intermediates, although it can be done. Another essential thing that's about the enzyme mechanism is for each N2 reduced, 1 hydrogen must be evolved. It's also true that H2 is a competitive inhibitor of N2 reduction, but hydrogen does not inhibit reduction of any other artificial substrate. You'll hear more about those probably in Marcus's talk. Another important observation is that in the presence of N2 and D2, there's N2 dependent HD formation. N2 does not bind, no substrate binds until multiple electrons are accumulated, but N2 is peculiar and it does not bind until 4 electrons, about 4, have been accumulated. Another important observation that we've made is that all substrates and inhibitors appear to bind at the same face of iron molybdenum cofactor. All even electron states are paramagnetic, which means they can be interrogated by EPR, and all odd electron states are diamagnetic, and so they are EPR silent. Finally, Brian Hoffman has shown in the past several years that the E4 state, which means the state of which 4 electrons have been accumulated, there are 2 bridging hydrides which are attached to iron atoms within this iron molybdenum cofactor. So let me go over now the current model that we have. And again, this is not carved in stone, and it's not, our particular position is not universally accepted. But the model that we have proposed, that's with Brian Hoffman and Lance Seefeld, does satisfy all of the experimental observation and has made predictions that we have tested and have sustained the model. So here is the beast, iron molybdenum cofactor, also called the M center, at which substrates bind and get reduced, and inhibitors bind as well. Now, so the orange here are irons, the yellow is sulfurs. There is a molybdenum, in the case of molybdenum nitrogenase, which is coordinated by homocitrate and also anchored to the protein through a sulfur here and a nitrogen ligand here. One thing that you'll notice is that this thing is all held together with a carbide. I'm not aware if there's any other carbides in biology that are involved in enzymology. This is a big debate now in the field is, what does this carbide do? Is it simply a heart of steel that holds these things together, holds the metal centers together, or is it a beating heart for which some of these bonds between the carbide and the irons at one of the faces breaks, allowing or shortens as a way to activate the binding site. Recent studies by Oliver Einsla and Doug Reese with really remarkable structures with this protein, or this metal cofactor contained within the protein, the X-ray structures, show that with CO bound, two molecules of CO inhibitor bound, there is no change in the distance and these bond lengths, which suggests it's a heart of steel. We've recently found and reported that we can replace this the carbide with C13, which allows the interrogation of these different bonds under turnover condition, or in the case we've just had accepted in JAKs, with a CO inhibitor bound. And our data so far suggests that it's, and agrees with the crystallography, that at least in the case of binding the inhibitor CO, that the carbide is a heart of steel and not a beating heart. So various experiments that we've done over the years has involved using genetic manipulation to substitute amino acids, which are in the first sphere of non-covalent interactions. So here you see this is this cofactor here turned towards you, and the homocitrate taken off so you can visually see what's going on. So what kinds of experiments we've done is we've substituted this histidine with a glutamine. And when we do that, we find that the enzyme can still bind N2, but it cannot reduce N2. And by binding N2, I mean it still acts as an inhibitor of all other substrate reductions. We've also substituted this glycine, which is very flexible here, with a serine. And in that case, it locks this valine into place. And under that condition, the enzyme, the modified enzyme, can reduce N2, but it's very poor at reducing acetylene. The other experiment we've done is to take this valine and substitute it with an alanine. And when we do that, the enzyme is able to reduce not only acetylene, but other longer chain hydrocarbons such as propylene and propargyl alcohol. Finally, what we've been able to do is substitute this valine with an isoleucine, so it blocks this phase. And in that case, what happens is the enzyme can no longer reduce any substrates except the reduction of protons. And so that gets us to the point of where we think then we've identified by these experiments and a few others that this is the action, at least for substrate activation. Now, there's a lot of talk these days about movement of the, once the a substrate is bound, that it might move. That's could happen, and that has been shown crystallographically by the Reese group using at least the inhibitor CO. Okay, so the model that we built all of this on is a model that was originally developed by David Lowe and Roger Thornley numerous years ago. And what this shows, E0 indicates the enzyme in its resting state with no electrons and no protons accumulated. And so it goes through a progression, and this is with each iron protein, molybdenum iron protein interaction, where one electron is delivered, 2 ATP are released. Then we go to the next step. This is EPR. This is diamagnetic. You can see it by EPR. This is paramagnetic. And this is diamagnetic. Now we get, once we have accumulated 2 electrons, if there's no other substrates available, this thing can go back, can evolve hydrogen and go backwards. And so you progress through the system, and at the E2H2 stage where 2 electrons and protons have been accumulated, or when you get E4, where 4 electrons and 4 protons have been accumulated, you can always go backwards. Now, at this E2H2 stage or E4H4 stage, you can use the artificial substrate, acetylene, and get this to go backwards. And so instead of evolving hydrogen, it will evolve ethylene. And if you add enough acetylene, it will completely suppress the ability to reduce hydrogen. Okay, this stage right here, E4H, is one that we were very interested in trapping, because this is the state where the Lowe and Thornley model suggested N2 is able to reversibly bind and get activated to the diazine level. Remember, this is the state which now overcomes the thermodynamic barrier. And so one of the things what we did is I isolated a protein for which the isoleucine was substituted here. So nobody can get in here except protons. So that means we've blocked the system. We blocked it right here. And we know that we had that E4H4 trapped because it has a distinguishing EPR signal. It's paramagnetic. And we knew it was that we were able to determine it was the E4H4 state because when we froze the sample and then thawed it very slowly, we could jump from here to a new EPR state here, and then thaw it a little bit more, and then go to this state. So Brian Hoffman then, using Endor spectroscopy, was able to elucidate that the E4H4 state, which was trapped by this amino acid substitution, the protein has, or the cofactor has two metal hydrides bridged between two iron atoms. And this led to what we call the reductive elimination model, where these hydrides are important for the activation of the system, such that during reductive elimination, 2 electrons are lost as hydrogen gas, and the N2 or the N2 molecule can bind and is promptly protonated by protons attached, we hypothesize, on the bridging sulfurs. Now, what is really important to know.
Dennis, I have a very stupid question.
Sure.
And I'm going to start here because I don't want to go on to the end. What allows N2, which is one of the most inert gases on the planet, that's not, how does it bind? What is binding?
Well, I'm not sure I know the answer to that question. I mean, there's, it could bind, either end on to one of these irons. So we've got 2 electrons that we're putting into the system. So in the words of Brian Hoffman is, one or more of these irons are red hot. And so when the hydrogen is evolved, you've got 2 electrons available, and there's also 2 protons available, and that's the activation. So that's the energy that you get that's needed to get this species. Exactly how it binds, where it binds, we don't know.
Thank you.
Okay.
Yeah, I'm just amazed that, we don't know.
Yeah. So the thing is, and one test of this model, of course, is if you can take H2, if this is reversible, if you then do this and you add D2 instead of H2, instead of hydrides here, you will get deuterides, and the system can go back, and that explains how HD is formed. So that's where we are in terms of what we think is the central feature of activation, is the use of metal hydrides to store multiple electrons that can be used to activate the system to get us to the diazine state.
Okay, thank you.
Now I want to move a little bit on, and this is just a diagram to show you how complicated the system is from a genetic perspective. So what you see here is a diagram of all the genes that we know are involved in nitrogen fixation in Azotobacter. They're directly involved in nitrogen fixation. And these involve catalytic components, metallocluster assembly components, trafficking components, electron transfer, regulation, oxygen protection, and unknown functions. Now, one of the giants in the field, in my opinion, is a person named Paul Bishop, who just using Petri plates and toothpicks was able, and changing the modification of the media, just an excellent microbiologist, was able to establish that not only are there components for a molybdenum nitrogenase, D and K encoding the molybdenum iron protein, H, the electron transfer reagent, but also there is a analogous system that can use vanadium instead of molybdenum as part of the cofactor, and that's included with H, D, G, and K. And then he went on to discover that not only are there two systems, there are three systems, and their third system contains neither vanadium nor molybdenum, but the cofactor is entirely constructed, as far as we know, with iron as the only metal. And so the question then is, what is the relationship? Do these enzymes all use the same mechanism? And that was a big question, because if we looked at the stoichiometry of the reactions, when we used the molybdenum system, it appeared that the minimum requirements was 16 ATP, 8 electrons, and 8 protons. But in the vanadium system, it took 24 ATP, 12 electrons, and 12 hydrogens. And for iron-only system, even worse. And so that raised the question, well, do these enzymes all work the same way? And the answer to that question is yes, they do. And because we've been able to isolate all three of them and compare them side by side, and we've been able to show that in the absence of all other substrates, electrons are diverted to hydrogen reduction for all three systems. For each N2 reduced, one hydrogen must be evolved for each system. H2 is a competitive inhibitor for all three. H2 does not inhibit reduction of other substrates. And in the presence of N2 and D2, there is HD formation. And so the question then is, well, what's the difference? And why is the iron system and the vanadium nitrogenase, why are they so much worse? And we examine that by looking at the effect of increasing the pressure of nitrogen gas. And you can see with the molybdenum system, it's at about 1 atmosphere. It works pretty well. And again, when at higher pressures, the best you can get is 1 N2 reduced per hydrogen released. And we also see the same thing with the iron nitrogenase and the vanadium nitrogenase, but at the physiological concentrations of N2, they are very wasteful in terms of evolution of non-productive hydrogen evolution. And this can be explained by measuring the rate of these reactions. And what we've been able to establish is the reductive elimination step for productive N2 binding is much slower for the alternative nitrogenases than for the others. Okay, now I want to move on quickly to another aspect. It's been known for many years that for the synthesis of at least the iron molybdenum cofactor, it's not assembled into by the apo form of the molybdenum iron protein stepwise, but rather is separately synthesized and then inserted. And so what we know now from the work of many people is there are two nodes involved in the assembly, the separate assembly of the iron molybdenum cofactor. One of those nodes is provided by NIF-B, which makes a precursor, which is basically the shell, the iron sulfur carbide shell with iron here. We call that precursor NIF-B-Co. Others call it the L cluster. Anyhow, one node is involved in synthesizing this factor we call NIF-B-Co, which I'll show you in a minute. And then it is transferred to a secondary node made up of NIF-E and N, where the homocitrate, where the iron, the apical iron is replaced with molybdenum, and the homocitrate becomes attached. And so the question is then do all of these three different systems operate the same way? And it seems likely in the case of the vanadium system, because the vanadium has analogs to the scaffold EN shown here for the molybdenum system, but the vanadium system has the same too, but the iron nitrogenase does not. And this is important from the practical perspective, because one of our goals is to put the simplest system we can build into plants so plants can make their own nitrogen fertilizer. So if we look at from a historical perspective, Paul Bishop has made the claim that the NIF-EN cofactor can functionally replace the BNF-EN cofactor, and that either the NIF-EN cofactor assembly scaffold can replace the BNF-EN assembly scaffold, and that either the NIF-EN or BNF-N scaffolds can be used to make the iron-iron cofactor. So what I want to show you now, that is not true. It's completely wrong. This is the NIF-B-Co, L cluster, if you will, that is synthesized through the function of NIF-B, which I'm sure Marcus will tell you more about. And what we've shown now, which I'll show you very quickly, this system, this cofactor is processed through a function of NIF-EN, probably trafficked by NIF-X. NIF-B makes the homocitrate from alpha-ketoglutarate and acetyl-CoA. So NIF-EN is absolutely necessary to make iron-limited cofactor, and it cannot be used to make iron vanadium cofactor, and it cannot be used to make the iron-iron cofactor. Furthermore, BNF-EN nor NIF-EN can be used to make the iron-iron cofactor. This is shown here. So we've made various mutations. This is Anna in the lab did most of this work. So if we knock out this gene, these genes, the molybdenum system, and we knock out the scaffolds for BNF-ENN, this is strain 2254. This is a control, it grows fine. And if we delete NIF-D and K and NIF-B-EN, it still grows. And this is in complete agreement with the original hypothesis. However, what they didn't test was why are these cells growing here? Are they using the vanadium system, or are they using the ANF system, the iron-only system? And the answer to that question is no, they're not using the vanadium system. They're using the alternative iron-only system, because when these genes are knocked out in combination with these, they can no longer grow. The conclusion being NIF-EN cannot replace the function of BNF-EN. And then to move on, the other experiments that we did is we knocked out, Anna knocked out these genes, the NIF-D and K for the molybdenum system, BNF-D, G, and K for the vanadium system, and then asked what happens if you knock out this scaffold for the molybdenum system, or this scaffold for the vanadium system, or both of them in combination. And the answer to that question is you don't need either one. All three of these different, four of these different combinations grow just fine with an active system. So none of these systems are required. This is also shown here where we purified the iron-only nitrogenase from strains that are intact for the other systems, or NIF-E is deleted, or BNF-EN is deleted, or both are deleted. And we're able to purify lots of protein in these genetic backgrounds. And this shows the activities for the various ones, and they're all perfectly fine. So what this means is that BNF, the completion of the iron-iron cofactor, unlike these other cofactors, is completed within its catalytic unit, the iron-iron protein itself, which is a big step in understanding from the practical perspective. It eliminates a step that we need to consider in terms of placing these genes in an active form into plants. Okay, the next thing I want to talk about is, so we have these different cofactors that are being made, and one of the questions we've been interested in is, again, what is the minimum unit we can use to make an iron-only nitrogenase? And one of the experiments that was performed, again in Paul Bishop's lab, is he found when he deleted this gene that's called ANFO, which is co-transcribed and with the structural components, when he deleted this gene, what he found was the protein that the cells could not reduce N2, but they could still reduce acetylene. And so what that suggested to us and to many people is that ANFO had some function in modifying the cofactor. And so we started to look into this. We're very excited about that possibility. And so this experiment is recapitulation of what was reported in the literature. When we delete ANFO, the cells cannot fix nitrogen. And so, what is going on here? And so we then grew up the cells. This is a strain that's deleted for ANFO. And then when we purified the protein, we found that it was completely the same as the wild type. So how could that be? How could you purify this protein in an ANFO deletion background for which the cells can't grow? And in this case, it's important to understand when we do these experiments, we grow the cells up to a very thick level before we switch them to a nitrogen-free media so we can induce the enzyme. And so what that suggested to us is that when we do this experiment, what we're doing is we're probably mopping up something in the system out of the media that is preventing the production of an active iron-only nitrogenase. And so the obvious possibilities is that it could be either vanadium or molybdenum that is causing this effect. And so to ask that question, what we decided to do is we asked if tungsten, which is an inhibitor of both molybdenum and vanadium uptake, can reverse the ANFO phenotype. And the answer to that question is yes. So here we have an ANFO mutant grown just in regular media. If we add 1 millimolar tungsten to that media, it can no longer grow, or now it can reverse the effect. So that then suggests that ANF phenotype is linked to vanadium acquisition. And so what we did then is the same experiment I told you a minute ago. We did this, grew the cells, but in this case, we added 5 micromolar of vanadium to the growth, and this is either to the wild type in terms of iron-only system or an ANFO deletion. And what we find is now we recapitulate the original observation that these proteins, the iron-only system, is completely incapable of reducing dinitrogen, but can still reduce acetylene and protons fairly well. There's also some inhibition of vanadium in the wild type for the iron-only, but it still has substantial activity. And so this pinpoints the association of vanadium as the inhibitory effect. And so we wondered whether there was going to be vanadium itself that caused this inhibition, or could it be that the BNF-EN cofactor, the BNF iron vanadium cofactor was sneaking into the iron-iron protein during its assembly and preventing the correct insertion of the appropriate iron-iron cofactor. And we were able to test that idea by asking if we could make a strain that, of course, we start with the ANFO deletion. Could we reverse that effect by deleting BNF-EN? And sure enough, we're able to do that. Then we looked at the protein purified from these two types of cells. And when we add vanadium to the system, in the absence of the deletion of BNF-EN, we get a large amount of vanadium incorporated into the protein. And if we do the same thing in a BNF-EN background, we get very, very little incorporation. So our conclusion is that ANFO phenotype is the consequence of misincorporation of iron vanadium cofactor into iron-iron protein. And what the ANFO's function is, it's a gatekeeper which prevents the misincorporation of the wrong cofactor. Again, this shows the activity data. Here, there's no activity in an ANFO mutant when grown in the presence of vanadium. And that is completely reversed biochemically when we also delete VNF-EN. So this is the take-home message of that part of my talk, which is that iron protein with iron-iron cofactor reduces N2, reduces acetylene, it reduces protons. But if in an ANFO deletion background, we get a hybrid protein which does not reduce N2 but does reduce the other substrates. And again, the importance of this, again, it's whittling down the number of genes that are needed in terms of transferring a robust nitrogen fixing system into incorporation into plants. And I'm going to close with that, just again to remind you how incredibly complex this system is, not only from the biochemical perspective, but also from the genetic perspective. And one of the goals we have in our lab, together with the collaborators that I mentioned to you, what is to figure out what all of these things do, how they're controlled, and what are the essential elements. And because we think that once we are able to get the correct genes, the minimum set of genes into making a nitrogen fixing eukaryote, it's going to be very important to optimize those so we achieve the goal, the nitrogen fixing dream of making nitrogen fixing plants. And with that, I'll close and answer any questions.
Thank you very, very much, Dennis. That was a great talk. We have several questions. I'll begin with, well, let's go back up a little bit here. Could it be that the carbide is enabling better geometry for coordination of the N2 between two irons as compared, for instance, to the geometry in the center of the P cluster?
Yeah, I think that's really a reasonable speculation. And that would go back to the idea that it's a heart of steel, which is holding everything in exactly the right, in the right geometric dimensions to get activation. So, so we're not claiming it's not involved in the activation. What we're claiming is it's not involved in the breaking of iron and carbon bonds during the activation.
All right. So the second question is from Banu, and he asks, what are the midpoint potentials for the P cluster, the M cluster, and for nitrogenase as a whole?
I'm going to leave it to Marcus to answer that question in his talk.
Very good.
He's more knowledgeable about those issues.
And so Saroj, who worked with Eric Boyd for his doctorate, asks, how would you recommend to assemble the M cluster and P cluster in vitro? And since the P cluster looks very similar to the M cluster, do you think that they can also fix N2, or might it have done so in the past?
So the question is, is it possible to activate substrates at the P cluster? There's
Right.
two different diverse opinions on that. My opinion is no. Now, from an evolutionary perspective, I think there's a great deal of information that's been published recently, but there's a lot of topological similarities in the formation of both the P cluster and the cofactor, which is dramatic. But as far as the evolutionary relationship of what evolved from what, I really have no opinion or grasp on that.
So fair enough. I'll just put some context here. The turnover time of nitrogen in the atmosphere is estimated to be 1 billion years, which suggests that before the evolution of oxygen, we had roughly between 0.9 and 1.1 bar of nitrogen, depending on how much CO2 was in the atmosphere, which we don't know very well. So the nitrogen in the atmosphere has been relatively constant over the last 4 billion years, actually. Why then do you think that if we have an evolutionary sequence, which Eric Boyd would suggest, and started with the molybdenum iron protein as the earliest one, why do you think we got these other two nitrogenases?
I think it's the most logical way to explain that is fluctuating of the availability of molybdenum in different microenvironments. And you've got this organism that's exquisitely designed to be able to switch to one system to the other based on the availability of the heterometal.
Yeah, so this is the problem. So in the early Earth, molybdenum was much less available than iron by probably about 3 orders of magnitude. So it just, from a point of geochemistry, doesn't make a lot of sense. I have one question that I'm going to ask you. What is the ATP doing?
The ATP, well, again, this is a matter of opinion. My opinion is the ATP is involved in the dissociation of the two components after electron transfer. So the two proteins dock and they undergo conformational changes. We know that because if you change the viscosity in the reaction milieu, you slow down the rate of electron transfer. By kinetic measurements, we know that electron transfer occurs much more quickly. Intercomponent electron transfer occurs much more quickly than ATP hydrolysis. So it's the energy of protein-protein association, which, in my opinion, drives electron transfer. And it's the energy of ATP hydrolysis, which then it dissociates the protein. Now, the important thing to remember here is we're trying to accumulate electrons unidirectionally. And so the way we think about this is ATP and protein-protein interaction dissociation provides an opening, a gate to provide electron transfer. And then when there's dissociation, it locks the system so the electrons don't go back the other way. And this occurs through multiple association and dissociation events.
All right, one last question from Amanda Garcia. It's wondering if the ANF system might give some hints as to how nitrogen-fixing organisms without NIF-EN genes might still assemble a functional NIF.
Yeah, we think, well, in my opinion, the ANF system is like a NIF-EN or a DNF-EN, and they're both, they both have they're structurally similar. We've known that for a long time, and they're very, very similarly organized. And it's just that the B cofactor, or L cluster, as it's sometimes called, is able to enter the protein without any accessory proteins, and so it acts as its own scaffold, which again is very appealing from the perspective of trying to make us the simplest system possible.
Yeah, so are you aware, there's a paper which was sent to me by Jim Olds, it's not published that I can tell, from a Chinese group at the Shanghai Academy of Agricultural Sciences that claim just a few weeks ago that they could insert nitrogenases into crops.
It's funny that you mentioned that. I had an hour-long interview with the New Yorker last week that also saw that article and wanted to know what's going on. And if you look at that, that's a preprint. If you look at their some of the experts in the field have completely demolished those claims.
Okay.
They're just outlandish in terms of what's claimed.
So that's what I told Jim as well.
Yeah.
So this goes back to David Valentine. Many other people have thought about this for many, many years. And I don't think we've gotten around the problem really, right?
That's correct. We're a lot further today than we were 40 years ago.
Right, when Valentine was working, right. But that was the dream from DOE. Dennis, thank you so much. It was really I really enjoyed this talk a lot.
Thank you.